Beyond preservation, could new research enable bone regeneration? For decades, the medical approach to treating osteoporosis has been defensive. Most current therapies focus on slowing the rate of bone loss, essentially acting as a brake to prevent further damage. However, a new frontier in medical research is shifting the focus from preservation to regeneration, the ability to actually rebuild bone that has already been lost. The mechanism of bone growth. To understand this breakthrough, one must look at the cellular level. Bone health is a constant balancing act between two types of cells: those that break bone down and osteoblasts, the cells responsible for building new bone tissue. Researchers have identified a specific antenna on the surface of these osteoblasts, known as the GPR133 receptor. This receptor acts as a communication hub, responding to two primary signals: physical stress, the mechanical pressure exerted on bones during activities like walking, running, or weightlifting; chemical signaling, a specific molecule called PTK7 that triggers the receptor to activate. By studying mice lacking this receptor, scientists confirmed its vital role. Without GPR133, bones become thin and fragile, mirroring the symptoms of osteoporosis in humans. From theory to testing, the AP503 breakthrough, the most significant finding in this study, involves a compound called AP503, which is designed to activate the GPR133 receptor. To test its efficacy, researchers used a model involving mice that had their ovaries removed, a standard scientific method, to simulate the rapid bone density loss women experience during menopause due to declining estrogen levels. The results were distinct from traditional treatments. Increased osteoblast activity. The compound stimulated the bone-building cells to work more effectively. Improved bone density. Rather than simply slowing the degradation process, the treatment appeared to actively increase bone mass. This distinction is critical. While current drugs aim to stop the leak of bone minerals, this new approach aims to refill the tank. Why this matters for women's health? The implications for women are profound. Postmenopausal bone loss is often rapid and difficult to reverse because the hormonal shift that triggers it is permanent. Current medical interventions can mitigate the damage, but they rarely restore the structural integrity lost during the transition into menopause. If therapies can successfully tap into the body's internal GPR133 signaling system, medicine could move toward regenerative treatments that help women reclaim bone strength rather than just managing their decline. Looking ahead, it is important to note that this research is currently in the preclinical phase. While the results in mice are highly promising, they do not yet guarantee the same success in humans. Significant time and rigorous clinical trials will be required to ensure safety and efficacy for human use. Until such therapies reach the market, the most effective way to support bone-building cells remains lifestyle-based. Weight-bearing exercise, strength training, and maintaining adequate levels of calcium and vitamin D conclusion. This research represents a potential paradigm shift in bone health, moving from a strategy of damage control to one of active regeneration. If successful in human trials, it could offer a transformative way to treat osteoporosis and support aging populations.